All right, so this is a little mini lecture to do with body fluid compartments. This is for the GLS station for week 5 for shock. So we'll just start with this diagram. Now imagine this is a full adult person. So 40% of your body weight is actually not water, it's minerals, protein, glycogen, fat. The rest of the 60% is completely water. So this is down here, 60% of your body weight is water. So we call this the total body water, or TBW. Now of this total body water, 2/3 or 40% of your whole body, 40% is intracellular water. So this is water that's within the cells, so your muscles, your nerves, the water that's in those cells is called your intracellular water. Then anything outside the cells is your extracellular water, or ECW. So that makes up about 20% of your body weight. And so what ECW is your interstitial fluid. So that's kind of like your lymphatic fluid, any fluid that's in between the cells. And a smaller part is your plasma, which is your intravascular volume. So the fluid that's in your vessels, that's your plasma, and that makes up about 4 to 5% of your total body weight. Now there's two membranes, two barriers. One is the cellular membrane between the ICW and ECW, so that divides the cells from the extracellular water. And then there's also a capillary membrane, which is your vessels, which divides the intravascular compartment and the extravascular compartment. So this is this. Now we've looked at the percentages. Now we're going to look at the actual volume. So let's say we have a 70 kilogram adult. So 60% of 70 is 42 liters. So there's about 42 liters of water, total body water, in an average 70 kilogram adult. So if we break that down, the 42 liters, 2/3 of it is in the cells, so that's 28 liters, and the other third is outside the cells, the extracellular fluid compartment. So if we break down the ECW, or extracellular water, again that's divided into the plasma. So that's the water within the vessels. And then the interstitial fluid down here, which is part of your extravascular fluid. Most of the extravascular fluid here is the interstitial. So these are kind of two and the same. All right, so now it gets interesting. So now we understand the different body compartments. How does this relate to septic shock and in the clinical setting of delivering fluid? So we'll come back to, I'll just go back to this diagram here. So if we think of fluids we give in the ED, we can kind of break them down into 3 groups. These are your crystalloids, which is your sodium chloride or saline, and there's things called Ringer's lactate or Hartmann's. All of those are called crystalloids because they have electrolytes in them. Then there's your colloids, things like albumin. And then there's dextrose or dextrose saline, which is another type of crystalloid, but that has sugar in it. Now if you look at this line, you can see dextrose filters all the way across the intracellular fluid and the extracellular fluid, whereas sodium is restricted. It can only stay in the extracellular compartment. It does not cross into the cells. Colloids remain just in the plasma. So colloids like albumin, the albumin protein is a very large molecule and it cannot cross over this capillary membrane. I'll just explain that again, but just with this diagram here. So imagine this is your IV line. You've put a needle in and it's accessing the veins and your plasma compartment. So let's give let's say some sodium chloride. So you've given some intravenous saline through here. Now, sodium chloride can distribute across the capillary membrane. It's a small molecule, it carries water with it, it goes into this plasma and the interstitial fluid. So it carries across the full 14 liters of the extracellular compartment. However, when saline, sodium chloride, hits the cell membrane, it cannot enter the cell, and that is because of a special protein, a sodium-potassium ATPase pump. Sodium-potassium, so NAK ATPase. That sits here on the cell membrane, and that spits out sodium back into the ECF in exchange for potassium. So potassium is high inside, but low outside the cells, whereas sodium is high in the plasma and low inside the cells. So sodium-potassium ATPase pump keeps normal saline within these two compartments. Let's say we give glucose, or say 5% glucose, 1 liter of 5% glucose or dextrose. We pump that in our IV line pump that into our IV line, into the blood. Now, glucose can travel across capillary membranes, but there's also special channels called GLUT receptors, G-L-U-T, and these allow glucose and water to freely pass into the intracellular compartment. So if you give 1 liter of glucose, it will come and distribute across the entire total body water volume. And that's different from normal saline, which just stays here in the extracellular compartment. What about colloids? So that's things like albumin, intravenous albumin. If you give albumin in here, the albumin molecule is so big, it cannot push out through the capillary membrane. So albumin ends up staying here within the plasma compartment. So in summary, if you give normal saline, it distributes across these two compartments. If you give dextrose, it goes all the way across here. And if you give albumin, albumin stays in the plasma. Why the hell is this important? Why do we care? Well, you may have heard of something called intracerebral edema, or cellular swelling of the brain. So that's why dextrose is dangerous to give straight. If you give dextrose, there's an increased risk of intracerebral edema because free water goes into the neurons and swells the cells. If you give saline, let's say we give 1 liter of saline, only a quarter of the saline will remain in the plasma, and the other 75% will distribute into the interstitial fluid. So that's why you can get peripheral edema when you give too much saline. As for albumin, if you give albumin, theoretically it stays here in the plasma compartment. So that's pretty much all I wanted to say about fluid compartments and the way different fluids distribute across the different compartments.